Hey everybody, welcome to Chapter 3, Parts 3.6/3.7, and this is going to focus on cell respiration. The original bunch of slides I set out were a little bit confusing, so I tried to tweak this. So if you watch this video, it'll be a slight bit different than the one that you have. A little bit more information, a little bit rearranged. So just flow with it and go with it. Okay, so we're going to start with cellular respiration. Now, cellular respiration can be aerobic, which is what we do in the mitochondria, but also there's a portion of it that could be anaerobic. So we're going to talk about both of those, and I'm going to start with that and work our way up into cellular respiration, aerobic. So the formula for aerobic cellular respiration, now that assumes that you are dealing with oxygen. So that's your aerobic, and that's what you do. So the formula is your glucose or your sugar, which you got from eating, or if you're a plant, you got it because you made it. There's the aerobic portion, that's the oxygen. That is going to be huge. That's going to be the electron acceptor, which you're going to see later on down the road. And the products, if you will, or the waste, are going to be CO2, which is going to go into your blood, and then you're going to breathe it out, water, and of course, usable energy in the form of ATP. Aerobic respiration makes about 36 ATP molecules total. About. So the sugar is oxidized, which means that it is losing the hydrogens. So that becomes the CO. There you got. And the oxygen is reduced. It is the oxygen that's picking up the hydrogen. So it's being reduced. So it's the opposite of photosynthesis. So how organisms use energy stored in macromolecules. Now, they do use glucose primarily, but they can use proteins and lipids. We're not going to talk about that here. We are going to focus on the glucose. So we got a couple of words here that you got to kind of get straight. The first one, and again, I'm sorry that it's a little confusing. So we're going to do either lactic acid or alcohol fermentation or cellular respiration to produce ATP. Now, lactic acid and alcohol fermentation are going to produce only 2 ATPs. Aerobic cellular respiration is around 36 ATPs. So you can see there's a very big difference. The original fermentation, the anaerobic respiration, is super primitive. It has been preserved and it has been passed down throughout geologic time, billions of years. It's simple. It's anaerobic, and it happens in the cytoplasm, of which all cells have. So we'll talk more about that as we move on. Eukaryotes, which would be you or plants that have mitochondria, it's going to be a coordinated enzyme catalyst reaction. So we'll talk about that as we go. Once again, it's primarily carbs, but they could use lipids and proteins if they have to. But we're going to focus on carbs only here. So, fermentation. Let's start with that because that's basic. We have lactic acid fermentation, that's one choice, and alcohol fermentation, which is a second choice. Okay? In each, only 2 ATPs are produced from one glucose. So one glucose, which is C6H12O6, makes only 2 ATPs. But what is actually more important in this process is the production of these molecules called NADP. That is what allows you to continually make ATPs from the glucose. Very important. Now we're going to see later on when we get to aerobic, it's a little bit different, but the key is to reproduce this NADP, and I'll show you what I'm talking about in a picture. So it's either going to, the end result to remake this NAD plus is going to be alcohol. So if you're in the yeast family, you're going to go on to make alcohol. And if you are in the bacteria family that's in yogurt, let's just say, you may go on to make lactic acid or lactate. And in human muscles, when we go anaerobic, which we'll see coming up, we will produce lactic acid as well, which is very painful the next day. So let's take a look here. There's your one glucose right there. There's your 2 ATPs that you net. Keep in mind, there's a little bit more to the story than this, but there's your net ATPs. Now, what you're going to find when we do aerobic respiration is it's this process called glycolysis, which is right here. Now, for us, for eukaryotes, for aerobic cellular respiration, these pyruvates are going to go off and do a bunch of good stuff. But if you are bacteria, you don't have that mitochondria to go on and do good stuff. So in order to continually make ATPs from glucose, you need these molecules of NADP. Because what happens is, if you keep on making NADH, then what? So you made your 2 pyruvates, big deal. But watch what happens. In lactic acid fermentation, these 2 pyruvates are going to go back and generate lactate. And that's what's going to be that kind of sour taste in yogurt. Remaking or regenerating NAD plus, which could then be used on the next glucose to make more ATP. Now, if you're a bacteria, you don't got a lot going on. You don't need a lot of energy. You're small. The stuff that's going on inside of you is not crazy difficult. So 2 ATPs from the glucose is fine. So if they're living on a nice medium of sugar, then bacteria will be fine, happy-go-lucky and dandy. Well, what happens if you're in the yeast family or you're part of what's in grapes? Or apples will do this as well. They have the glucose, which is fine. They're grapes and they're apples. They got plenty of sugar. They're going to go through a glycolysis. They're going to produce their 2 ATPs. This is anaerobic. So if you want to make wine or you want to make cider, hard cider, you have to take these grapes and you have to crush them down, turn them into juice, and then lock them up in a sealed container where they don't have access to oxygen. So what they're going to do is they're going to produce the ATPs, so they're very happy they're growing. There's your pyruvates. Everything's cool. Sounds just like lactic acid. But there's one more step here. You're going to get rid of CO2s. So in things like beer and in things like champagne, those are the bubbles. You're going to wrap around, once again, regenerating your NAD+, so you can go on to make more ATPs from the glucose, and there's your ethanol or your alcohol. So those are fermentation that happens in prokaryotes and some eukaryotes if they go anaerobic. So if you see here, lactic acid is going to happen in bacteria and in some animal cells. Again, we'll talk about a little bit. So who does what? Well, the bacteria in yogurt are going to produce the lactate or the lactic acid, and yeast will go on to make alcohol in beer and bread. So a lot of you then may be saying to yourselves, well, how come I don't get drunk if I eat bread? Well, what happens is the heat will burn off the alcohol when it's cooking, and the CO2 produced is what puts all those air pockets in the bread. So hopefully that'll make sense. And again, aerobic eukaryotes, like us, will go into lactic acid fermentation in times of low oxygen. Now, obviously, if you have no oxygen, you're going to die. But if you are really exercising excruciatingly hard, a lot, locally, those cells are going to go anaerobic. You're personally breathing still, so you're not going to die. But those cells, let's say in your legs, and if you're running a marathon or something, they are going to go anaerobic for a little bit and cause or form lactic acids. And you'll feel it tomorrow because it hurts. Okay, so let's go back to glycolysis now. So glycolysis is going to happen in every single organism on Earth. So we just talked about the first half of it that went into fermentation, but now let's kind of get a little bit more detailed. You're going to see it from simple bacteria all the way up to the cells of your brain. What it does is it takes the glucose, one. It's supposed to be one. One glucose into 2 pyruvates. Now remember, if you are bacteria or you are yeast and you're going to go anaerobic, those pyruvates are going to go to form either lactic acid or alcohol. But if those 2 pyruvate molecules are going to get altered and enter into the mitochondria. So again, we'll see that in a little bit. So the process of glycolysis, as a review, is considered primitive. Why? Because it occurs without the input of oxygen, because there was no oxygen back in the early atmosphere, and it happens in the cytoplasm, and we know that all cells have cytoplasm. And it has been conserved for billions of years as it's been passed down from pretty much every organism. So glycolysis does require the input of 2 ATPs. That was not on the last diagram that I showed you. And it's going to kick out 4. So you're like, well, wait a second. That diagram only had 2. Because it took into account there's a net gain of 2. So they're not talking about the 2 in, 4 out. They're just talking about the total net production of 2 and 2 NADH. Now again, for you, those NADH molecules are going to become important later, but let's see what we got. So what's next? In bacteria and many other cells, it's the end of the road. The pyruvate will go on to form alcohol lactic acid, which we talked about. We're regenerating NAD plus, and we're producing ATP, and that cell is fine. But if you are a eukaryote, you have mitochondria, you are now going to start the next phase. We're going to get the Krebs cycle involved. We're going to get the electron transport chain involved. We're going to form a molecule called acetyl-CoA. We're going to get that involved. So let's see where we're at. So the mitochondria itself, it's they draw it, kind of looks like a sausage. But what it is, it's a double membrane structure. It's got an outer membrane like that. It's got an inner membrane like that. It's going to have a couple of spaces. That's going to be called the intermembrane space. And that is going to be called the matrix. And a lot of stuff is going to happen in both of those, but we'll see that as we go. The inner membrane is highly folded. So you're going to see I drew it like that, highly folded. And the area inside is known as the matrix, which we said. And embedded in that membrane is going to be a series of enzymes and proteins known as the electron transport chain, otherwise known as the ETC. And we'll see that in a little bit more detail as we go. Just like in chloroplasts, mitochondria are going to contain DNA, enzymes, and ribosomes. So that kind of folds us back into the end of the last chapter with the endosymbiotic theory. Like, really? Is it possible? So again, just like chloroplasts, which we talked about in the beginning of this chapter, mitochondria, DNA, enzymes, and ribosomes. So there's a picture, a better picture, as you can see. So the cristae is that folded inner membrane. Sometimes they'll say the inner membrane, or they'll call it the cristae. And there's your, again, inner membrane. So point to the same thing. There's your outer membrane. You're going to have this really cool thing called ATP synthase, which we're going to talk about in a little bit. There's your DNA, which you care about. There's your ribosomes, which you care about. The word matrix, which you care about. And the intermembrane space. All of those things are going to become very important right now. So what is the electron transport chain? And hopefully we drew it enough times that it is basically a series of proteins and pumps and ATP synthase, and it has a bunch of jobs. So let's see what they are. The ETC is, again, a series of coupled reactions that is looking to establish something known as a proton gradient. In order to pump these protons against their concentration gradient, you're going against it first. You need to pump, and you're going to pump with energy from electrons. What? Crazy. Again, we'll talk about it. You're going to pump these hydrogens. They're going to build up in that intermembrane space. There's going to be a lot of them there. You just keep on pumping and pumping these protons. You eventually have so many that the mitochondria is like ready to burst. You're like, no, don't burst. So what's going to happen is that buildup of protons that you've now built up are going to come through this ATP synthase and produce ATP. Again, we're going to see this coming up in a little bit more detail. It's called chemiosmosis, because osmosis means water moving from high to low concentration. This is chemiosmosis, so it's going to be hydrogen ions. So this diagram, it zooms in. It's kind of hard to see, but here's what we got. We're going to find out in a second that once that pyruvate that we made in glycolysis, it's going to enter into what is known as the Krebs cycle. And it's going to produce these things called NADH and FADH2. Basically what these are electron carriers. And watch what they do. the NADH is going to come to the party right here. It's going to drop off its electron. It's now down to an NAD+. Oh, wait, that sounds familiar. Didn't we see that already? Now watch what happens. That electron is going to be kind of like a jumping bean. And what it's going to do is it's going to burn through that protein as 1, and it's going to burn through that one, which is 2, and then it's going to burn through that one, which is 3. Every time the energy from that electron passes through each of those pumps, it can move 1 hydrogen. So let's see what happens. There's a hydrogen, and you just moved it into that intermembrane space. Well, you move that one, and you move that one, and you move that one. Now you just move 3. So life is great, right? Well, what's happening is you're starting to get a buildup. Because think about it, and let's spell that, right? Starting to get a buildup. Now keep in mind, I'm going to be making a lot of these NADHs. You're going to see coming up, make a lot of them. Well, FADH2 is the same thing, but instead of passing through 3 pumps, it only passes through 1, 2. But nonetheless, is it still pumping hydrogens? Answer, yes. Now remember that oxygen that we breathe? There it is right there. The oxygen is known as the final electron acceptor. So watch what's happening here. The electron, as you can see, is zipping on through, zipping on, zipping on, zipping on, and boom. When it slams into that oxygen, and you have some H's kind of laying around here. Don't think that we pumped them all out. So you combine all those H pluses with all those electrons, and there is your waste product of water. So in with the oxygen and out with the water. So that accounts for a couple of them. We accounted for the glucose already. That got broken down in the glycolysis step. And the last thing is this thing called ATP. So in this intermembrane space now, we have a lot of these hydrogens. Lots and lots and lots. So, well, what happens? Stuff wants to move from high concentration to low concentration. But if you remember, our friends, the lipid bilayer, if you remember that from the old days, the lipid bilayer doesn't let charged things through. So they need help. So they're going to come through this ATP synthase. So it's actually facilitated diffusion, meaning the hydrogens are going from a high concentration to a low concentration, but they can't get through by themselves. They need help. Well, good news. Here's this ATP synthase is like, yo, I'll help. And in kind of return, I guess, as the hydrogen comes zipping through, you can see it zipping through right there, it has a tremendous amount of energy. And it has the ability to take that ADP molecule, add a phosphate to it, and guess what you just made? ATP. So think about it. Every time an NADH comes up to the ETC, it moves 3 hydrogens. Well, every time each of those hydrogens comes through, you just make an ATP. So you make a lot of ATP from all of these NADHs and FADH2s. So let's see what's going on. Unlike photosynthesis, where the electrons came from the water, if you remember, here the electrons come from the NADH and the FADH2, which again are going to be made, which you didn't learn yet, I'm going to tell you in a second, came from the Krebs cycle. And the electrons are going to be passed through these pumps, which we just said. Since the NADH goes through 3 pumps, that means it moves 3 hydrogens. And for every 1 NADH, you're going to have enough energy to generate 3 ATPs. Because remember, it goes through 3 pumps. The FADH2 only goes through 2 pumps. So for every FADH2, that's going to net you 2 ATPs. Now you're going to see in the Krebs cycle coming up, you make a lot of NADH. So there's a lot of ATP being made here. Now, in the Krebs cycle, they do make a tiny bit of ATP during the Krebs cycle, but the bigger deal is this NADH and FADH2. So there's our friend Krebs cycle. Now, Krebs cycle, it's also known as the citric acid cycle, which you may see. This is going to be found in the matrix. So when we put the whole story together, I'll show you. Now, what the major big deal here is, NADH that's produced, and there's the CO2 that we release. There's another NADH, more CO2 we release. There's an FADH2. There's an NADH. There's actually an NADH that's going to be formed over here too. And what happens is the pyruvate that was made during glycolysis is going to actually form into what is known as an acetyl-CoA or acetyl-CoA, however you feel like saying it. That acetyl-CoA can enter the Krebs cycle. The CoA gets kicked out, and then you join the cycle, and it just goes around and around and around. So the key here is you're losing CO2s. Great, that's fine. We're just getting rid of them anyway. We're making NADHs, and remember I told you the Krebs cycle does make a little teeny-weeny bit of GTP, which as far as you're concerned is the same thing as ATP. Make it a little bit. So far, in glycolysis we made a couple of ATPs. In the Krebs cycle we made a couple of ATPs. And we generated molecules that are going to eventually make a lot of ATPs. Here's a zoom in again of the electron transport chain. There's our NADH, and you can see there's a hydrogen that gets freed up. That hydrogen may be that hydrogen that's going to eventually go on to make our friend water, which is right there. You can see here how the NADH goes through 1, 2, 3 pumps. The FADH2 only goes through 2 pumps, but either way, we're moving hydrogens. Now, it's not on here, but if the ATP synthase was here, it would be right there, and those hydrogens are going to come screaming through there, and they're going to take ADP and turn it into ATP. So very, very important what's going on there. All the while, this is generating heat. That's another waste product. So that's why we do so much energy in our muscles, especially, is released as heat. So if you are endothermic, which would be you are going to be in the mammals. You're not a bird, but birds do it too. When we produce all this ATP, that byproduct of heat is important, and we hold it in, or most animals with fur do. They hold it in with their fur, and birds hold it in with their feathers. We wear clothes. So it's kind of how it all works out. Now, how is this similar? How is it different from photosynthesis? Well, both, if you remember, rely on a pump system. Remember the photosystems from photosynthesis? They are both dealing with building up hydrogen ions. They are both dealing with this ATP synthase. Now, what they do with their ATP is different, but it's still being made in basically the same idea how it comes running through the ATP synthase in a process known as chemiosmosis. In respiration, it is known as oxidative phosphorylation. It's called oxidative because the oxygen drives the party. In photosynthesis, it's known as photophosphorylation because the sun drives the whole party. So if we go back to the picture here, the oxygen right there is the driving force. In photosynthesis, the sun is the driving force. Okay? Now, what if you're a prokaryote? Well, you don't have any mitochondria. So they actually, believe it or not, have a small electron transport chain within their plasma membrane. Now, if you are aerobic bacteria, which some of them are, you can use that oxygen to help you. But in the world of anaerobic bacteria, they're going to, figure out other ways around it. The chance of you seeing anything like this is slim. But if you kind of look at the picture here, let's see, we got our ATP synthase, we got hydrogens, we got pumps, we got it all going on there. But again, I can't imagine they're going to get too deep into the prokaryotes. There's only going to be 2 questions on your test, so really how much can they get into it? So now, let's get specific. Let's kind of put it all together. Process of respiration always begins with glycolysis in all organisms. Everybody, you, bacteria. Prokaryotes, you have a net production of 2 ATPs. You're going to make 2 pyruvates in the event that you are, if you are bacteria, you're going to regenerate. This is NAD+. I made a mistake here. You're going to regenerate NAD+, and you're going to either make your lactate or your alcohol, depending. This process can be done without oxygen. Once again, it is anaerobic and it happens in the cytoplasm. So that is everybody, even you. It's called anaerobic respiration. Now, we don't need to do this part here. We don't need to do that part. We're going to do that later. But if we have to, we can do it. Again, refresh our memory. There's our glycolysis. There's our glycolysis. There's our production of ATP. There's our production of ATP. There's our regeneration of NAD+. And we either make alcohol or lactate. That's bacteria, yeast. You, aerobic. Say it again. You have mitochondria. It starts with glycolysis. Okay, nothing changes. In the cytoplasm, nothing changes. You're making ATP. Nothing changes. You're making pyruvates. Nothing changes. But now, the pyruvates have to get into the mitochondria now. Because remember, if that's the cell, and that's the mitochondria over there, glycolysis happens in the cytoplasm, which produces that pyruvate. That pyruvate has to get into the mitochondria. Well, the only way to do that is you're going to have to turn it into acetyl-CoA, which again we saw in the Krebs cycle. And you're going to produce your CO2. There it is. We're going to transfer some electrons, and we're going to make a lot of ATP. So that's aerobic, and we saw all the steps before. Review. In the cytosol or cytoplasm, you're making 2 ATPs. This is known as substrate level phosphorylation because you're literally just putting the phosphate on the ADP. Then you got to take that pyruvate. It's going to turn into acetyl-CoA. That's now going to join the Krebs or citric acid cycle. There's your NADH and your FADH2, which you're going to make tons of, which are going to go on to the ETC. There's our chemiosmosis we talked about. There's our lots and lots and lots of ATP. This was oxidative because it's driven by oxygen. Those little ATPs that are made in the Krebs cycle or the citric acid cycle, they're also substrate level. So that's simple. So notice, substrate level does not make a lot of ATP. Oxidative makes a huge amount. So if you add it all up, get about 38 ATPs. So it's a lot. Most books will be happy with 30 to 36, and that's fine. Again, why is this so useful? Well, it's huge because this ATP is going to be usable energy. Now, that usable energy is going to be important for everything. It's going to help you make enzymes. It's going to help move the chromatids in cell division. It's going to do everything. That's why you need it. So the generation of ATP is very, very, very important. Got a couple of videos, as always. You got the Bozeman and you got a cell respiration. It's kind of like an animation video. Both excellent. Now, what about fitness? Now, again, I kind of flipped the order here, so you're going to notice, well, where's the questions? They'll come up in a second. So, how do organisms use variations within the cell to survive? Well, of course, what does variation always do? Allows organisms to respond to a variety of environmental factors. So some of the things we may see in some cells is a variation in the phospholipids, which may or may not let certain things in, which may or may not deal with various temperatures. Different types of hemoglobin, let's just say, may be used in different kinds of organisms. Certain, like a whale, has a different setup than a human does. And in the world of plants, the different kinds of chlorophylls will deal with different wavelengths of light depending on the availability. So there's a lot of variation that's going to help cells survive. So let's take a look at our FRQs. We got 4 parts today. So 3 steps in aerobic cell respiration. So real quick, that's glycolysis, the Krebs cycle, and the ETC. Many of you did well on that. We'll get to the lactic acid in a second. I changed the question and knocked it down to 2 reasons because the 3 was a little kind of weird. But if you got the 2, I gave you full credit. And also part D, which, again, a lot of people kind of mess this up. And it's annoying. And every once in a while they'll ask it. And I would absolutely, I think they're going to do it this year for you. They want you to literally ask a question. They want a question. They're looking for it. You ask it. How does your question, even if your question has no good answer, it doesn't matter. They're looking for your question. So the three main steps in the aerobic process we said are glycolysis, Krebs cycle, and the ETC. And I went into a little bit more detail than you needed to get into here, but as long as you mentioned something along the lines of glycolysis produces some ATP, if you went in to talk about how the Krebs cycle produces some ATP, and if you went in to talk about the electron transport chain and you mentioned chemiosmosis and you mentioned ATP synthase, you got, again, about 30 more ATPs. It's a big deal. So as long as you mention each of those three and how they contributed, you got full credit on that part. Part B, almost every one of you got this right. I don't know if you looked it up or you just knew it, which is great. But lactic acid is going to build up in our muscles when there's a lack of oxygen. Once again, that doesn't mean that you're going to die. It means locally those cells are in a little bit of oxygen debt. And the byproduct, of course, is lactic acid. And tomorrow, that is going to hurt. But you didn't need to say that. Part C. Again, I kind of knocked it down a little bit. If you said that it was primitive and it was passed down, that was great. That was awesome. That was one. But the things I was looking for were anaerobic, a.k.a. without oxygen, and that all cells have cytoplasm. You could have also said that it happened outside of a membrane-bound organelle, which would have been the same exact thing. So as long as you said something like that, you got full credit. And last, again, this question was tricky, so as long as your question was decent, you were good. So here, can the enzymes found in the matrix of the mitochondria today have similar structure and functions to those found in prokaryotes? I don't know, but guess what it is? It is a question. So this was definitely challenging. This we did many, many, many months ago. So details, again, how detailed are they going to get? I don't know. But know the steps and know what happens if there's a mistake or an alteration in any of these steps. And if you remember from the question I asked on photosynthesis on how weed killers and plant killers work, is if you disable any part of the process, it doesn't work. And after you're unable to generate ATP, your organism's going to die. So you're looking for, if they ask a question on cell respiration and or photosynthesis, you're looking for what could alter it, because I doubt they're going to ask super specific stuff like this, but it's good to know. So again, hopefully that was helpful, and I'll see you next time.